All right. So last time we start talking about the cells, we talked about this cell theory, and we talked about the plasma membrane from outside, the nucleus from inside, and I'm sorry, this is the nucleus. The membrane from outside. Let me show you the whole thing like this. This one right here. This is the whole entire cell. Look at the blue. We talked about the cell membrane with the different details. We talked about this, which is the nucleus, which is the slide here. And in between, we called it the cytoplasm. And the cytoplasm is two parts: cytosol, which is the liquid part or the fluid part, and the organelles that we are going to talk about. We talked about the nucleus and nucleolus inside of it. And then we're going to move and talk about different organelles, right? So just to refresh your information, if you take, let me make it big again. Look at this one at the center here. This is the nucleus, and this is a magnified picture of the nucleus. This is what we're talking about right now. So this is the nucleus right here. We talked about the nuclear membrane. It's also a double layer of phospholipid, just like, or a lipid double layer, just like the blue here, which is the cell membrane. And inside, the inside here is called nucleoplasm. And the nucleus of the nucleus, this is not science. I'm just giving you a way to understand what does it mean. But it's called nucleolus. It's inside the nucleus. And I mentioned that it is where we make the ribosomes. Every single word that I'm saying is important. So what's a nucleolus, which contain LEO, not the same as nucleus, which is a whole thing. This is where we make the ribosomes. And in between in the nucleoplasm, this is where we store the DNA in eukaryotes. Remember, in the prokaryotes, we also have DNA, but it's not inside the nucleus like this. So this is where we have the DNA. So this is eukaryotes. Do you mean the DNA is just scattered all over right here? No, it's not scattered. It is arranged in the form of chromosomes and chromatin. We will discuss more details about it. It's right here. Chromatin is DNA and proteins. So is the DNA scattered in the nucleus, in the eukaryotes? No, it is in the nucleus, but not scattered. It is combined with proteins, and we call that chromatin. We said that we have pores, and these pores are tiny, small pores that allow only small things to go in and out, not the DNA. The DNA is too big. The DNA, as compared to these pores, is like a truck compared to the door of your house. You cannot go through, but you can take things from the truck and you move it out if it fits your door. Like you can take like a chair out of your truck and move it in through your door. That's okay. Applying that here, the DNA is like the truck or chromatin in the form of chromatin. This is like a truck that you cannot move out. It's too big. It doesn't fit in to leave. And this is intentional. It's extremely important. It contains our genes. It contains the original script. And it has to be kept within this big office, secured. It's too big to leave, but you can make a copy of the part of the instructions that you need. And this copy is the one that is small enough to leave through the pores. And we call that messenger RNA. We will talk about it later. Just wanted to give you an idea. I also want you to notice something here. Look at this surrounding the nucleus. You will see this organelle that's coming. We'll talk about it. You see how it's like a membrane that's folding into a network, and see how there are things scattered on its surface. This is basically surrounding here. If you make this picture bigger again to show you what I mean, look at this nucleus. Look at this membrane surrounding it. It's in the form of network. A network in Greek is reticulum. Reticulum means network. I advised you guys several times, Latin and Greek is prefixes and suffixes. Put things together, it will make a meaning. Okay, you can translate even if you don't know what it means. If you know the prefixes and suffixes, if you know enough, if you have no idea about a word, you can cut it into different pieces and translate with no prior knowledge, even if you don't know. So here it is. This is a network, all right, surrounding and attaching to the nucleus. So when something leaves the nucleus, it will go to this network. What's network in Greek? Reticulum. And where is that reticulum? In the cytoplasm. What do you want to call it? Endoplasmic reticulum. We will talk about it. This is what we talked about already. This is the nucleus with a double layer, just like the cell, 2 layers of phospholipid. There is an inner layer, an outer layer, and there is a space in between. This is the nucleolus, which is within the nucleus. This is where we make the ribosomes. In between, the space in between, it's called nucleoplasm. Translation, nucleus. Plasm is the medium. Nucleoplasm, the medium in the nucleus, which is this space. What do we have here? DNA. Is it just random scattered? No, it is not. It is combined with proteins. We call it chromatin. Look at these ribosomes scatter on the surface. Look at the pores and so on. Now, here is the part that I was just talking about. What is surrounding and attaching to the nucleus that we already talked about, and it's also connected to it. This is the endoplasmic reticulum I was just talking about. Endoplasmic reticulum. Translation, endo means inside. Plasmic, referring to the cytoplasm, this medium of the cells, this blue medium of the cells here, which is located between the nucleus and the membrane. So we call it endoplasmic reticulum, and you know now where does the name came from. There are 2 parts that you need to know, 2 types, I mean. One of them is rough, and the other one is smooth. What does it mean? It means if this reticulum or network doesn't have anything on the surface will look smooth, and we call that smooth endoplasmic reticulum. If there are ribosomes stuck on the surface looks rough because of these ribosomes on its surface. And if that's the case, we call it rough endoplasmic reticulum. So we have 2 types of this network in the cytoplasm. That's called endoplasmic reticulum, rough and smooth. So this is an organelle that we will discuss, 2 organelles, I mean, rough and smooth endoplasmic reticulum. What's the other organelles that we're going to talk about? Golgi apparatus, which is this one right here, vesicles, and others. We'll talk about many different things. But I started with those because these are called endomembrane. These are the ones that are surrounded by membranes. Nucleus is surrounded by a membrane, the Golgi apparatus, the vesicles, and so on. But anyways, here is the most important that's coming. Endoplasmic reticulum. What do we need to know about this organelle? There is rough and there is smooth. What's the difference? Smooth, nothing on the surface, and that's why the surface looks smooth. Function that you will be asked about. Synthesize, which mean form, and transport, lipids. Very important. This is a function. What does it mean and what does it do? These are the questions. Rough endoplasmic reticulum. What does it mean? Rough. What do you mean by rough? Ribosomes on its surface. What's a function? Is it also making and transporting lipids? No, it is not. This is different. Because of the ribosomes on its surface, it looks rough. And what's the function of the ribosomes? Making protein synthesis. So what's the function of the rough endoplasmic reticulum? Making protein, Does it make protein by itself? No, by the ribosomes on it, but it is going to transport it. Okay. So this is the smooth and rough, and very important. What does it mean? What does it do? These are the, so we got the nucleus, nucleolus, that's two. We got the two types of endoplasmic reticulum. These are 4 organelles inside the nucleus so far. And
Can
we
I ask a
talked about the
question?
membrane already. Yes,
What
go ahead.
causes, if you have a smooth ER, can a smooth ER turn into a rough ER?
No, it doesn't turn. When we were like embryo and fetus, it's already made. So this part is already smooth and this part is already rough. So this is how we were born. How did it form exactly while we were fetus? This is a little bit deeper. We don't need to know. But if I take any of our cells, it's already part of it smooth and part of it is rough. Okay. All right. Or you still have a question?
Nope, that answered it. I just wanted to know if smooth could turn into rough by
Yes,
things
it doesn't
attaching
turn.
to it.
It's already smooth, already rough.
Okay, thank you.
You're welcome. All right. The next one is Golgi apparatus, the next organelle. What's a Golgi apparatus or Golgi body? This body right here, it's like folded, like a folded membrane. And what we have to know here is inside this folding membrane that's called the Golgi body, there are enzymes that are going to take the proteins. Who made the proteins? I just told you. Ribosomes on the rough endoplasmic reticulum. It makes proteins. And then it leave it and go to this Golgi apparatus or Golgi body. Why do you send it to the Golgi body? Because the protein that we make in the ribosomes, in the rough endoplasmic reticulum, is not ready to use. It's raw protein. It's not ready to use yet. Okay, so if you like have some, if you make like flour, if you make flour, you're not gonna eat it. It's not ready to use yet. So you need to modify it first. So it goes to the Golgi body in order to modify the proteins to be ready for using. How is it ready to use? Yes, because Golgi apparatus or Golgi body is going to make some modifications in order to export it out of the cells. If this protein is needed outside of the cells, we are going to export it out. But before you export it, you have to do the finishing, the modification. And if you compare that, I always try to compare it to a factory. If this was a factory, and let's say the rough endoplasmic reticulum, instead of making ribosomes, it's making TVs. So the TV that we make it here is not ready yet. It's not completely ready. It's just parts that put together, and then when we send it to the finishing deck or the finishing body, you are going to take the TVs and you're going to like paint it. You're going to like add different things to it to look nice. If there is something bad, you take it off, and then you package it. You put it like you put foam surrounding it, and you put it in a nice box, and then you tape it together. Now it is ready to be exported out, and it goes from the factory to the different like stores to be sold. Did you ever go like to a store and buying something that's brand new, and they just give you like TV, here you go, take it? Of course not. It has to be nicely packaged. It has to look nice. There is foam around it. It's inside a box that's sealed. This is what the Golgi body does. After that, when it box it, make it like into boxes to be exported. The boxes itself are called the vesicles. I'm comparing what's happening to a factory. So after you do it, how can you export it? I put it inside nice boxes, I seal it, and these boxes will be taken out to be used out. So what do you call these boxes here as if you compare it to the cells? vesicles. It's parts of the Golgi body that's detached, carrying the protein after it's been finished, put nicely into these vesicles, and these vesicles are going to travel and leave the cells and go where it can be used. How about if you want to make protein for your own use at cells, not for export, for domestic use? Yes, it can leave and just go inside the cells, but you don't have to put it in these vesicles. You don't have to, it's for own use. Like if people working in a factory, they want TV, they will tell them just take a nice TV that you want and just take it. It doesn't have to be within the boxes and sealed, just as an example. And these vesicles that I'm talking about are different types. Some of them are called exporting ones. This is the one that's going to leave. But there are other types that also the Golgi body does, not just those that carry the protein out. There are others that are called lysosomes and peroxisomes. How do we understand this? Lyso and some is two parts. Lyso is coming from lysis, breakdown. Some is body. So these are the bodies that will break down. Simple. How does it break down? If there is something that you don't need, I'm not talking about exporting the protein now. These are the export vesicles. What else does the Golgi body does beside that? Besides finishing and modifying the protein and put it in these exporting vesicles. This is one type. Other types of vesicles. Does Golgi body does other vesicles beside the one that carry the protein to be exported? Yes, it makes another vesicles, lysosomes. They contain digestive enzymes. So if there is, it's like the recycle pen. If there is something that you don't need, send it to these vesicles, lysosomes, and these vesicles will contain enzyme that will dissolve, digest, break down the macro molecules that you don't need. Macro is big. Molecules. The big molecules send it to these lysosomes, and they are going to break it down, translation, lysis. And that's why we call it lysosomes. Look at this, lysosomes, prefix, suffix. I'm going to use the same suffix again. Look at this. And you're going to use all the prefixes and suffixes all the time, Peroxisomes, some is body, just like this, same suffix. What's peroxy? It's coming from hydrogen peroxide. Hydrogen peroxide is another one that's going to break down, you break down things into hydrogen peroxide, and hydrogen peroxide will help you to break down other unwanted materials. Depend on the nature, you can use both of those. So both of those are like the recycle bin. You can recycle something here, you can recycle something somewhere else. Like there is something that you can shred using the shredder, and there is other things that you can recycle by like you can put stuff in the trash, and some stuff you can put it in the recycle. So you recycle it in two different ways. Break it down. So this is just reviewing what we talked about. Nucleus, ribosomes, they contain DNA, and this is where it's stored in the eukaryotes, in the plasmic reticulum. Yes, there is rough and smooth. It's enclose or surround the nuclear envelope or work as a nuclear envelope. The ribosomes can be smooth or rough. We have nucleus, we have ribosomes, but which one of them make the or contain the DNA? Which one of those is the organelle that contain the DNA? Nucleus or ribosomes? What do you guys think?
Ribosomes.
Ribosomes as the one that contain the DNA?
Nucleus?
It's
Nucleus.
the nucleus. Yes, nucleus is the one that makes the DNA. Endoplasmic reticulum or nuclear envelope is the one that surrounds the nucleus. It's
Nuclear
nuclear.
envelope.
Yes, nuclear envelope is the same as membrane. Envelope or membrane. Okay, ribosomes can be smooth, can be rough. Yes, but which one make proteins? Smooth or rough?
The rough?
The rough make proteins and the smooth make lipids.
Lipids.
Yes, these are the questions that you're going to see. You have to know that, okay? Which one includes the nucleus, envelope, or membrane? Which one make proteins? Rough. Which one make lipid? Smooth. Which one break down here? Which one break down macromolecules, the big ones?
Peroxisomes.
It's right here. Look at this. Lysosomes break down macro, and peroxisomes break down unwanted material. Here it is. Lysosomes break down, and the peroxisomes unwanted material. Okay. Next organelle is mitochondrion. Mitochondrion is a symbol for mitochondria. In Greek or Latin, if you put ION at the end, it is single. If you put IA instead of ION, it is plural. So we're talking about one of them here. This is the powerhouse of the cell. You need to remember this word. What do you mean by powerhouse? This is where we make the ATP from organic compounds using oxygen. You should remember all of this. You can't just say, mitochondria make ATP from the food. We break down the food or the organic molecules and we make ATP. No, that's not enough. You have to have oxygen. Mitochondrion does not work without oxygen. You have to remember it. And here is how it looks like.
What does ATP stand for?
Oh, adenosine triphosphate. ATP is adenosine triphosphate. This is how we store and use our energy. Okay, it's coming. We will talk about it more. But for now, it's adenosine triphosphate, and this is the compound that's in which we make our energy. This is how we store the energy. All right. So here is the mitochondria. It looks like bean shaped like this. And there is a membrane outside. And there is another membrane, folding membrane inside. Two membranes. The inner one, the folding, is called cistern or cristae. In this case, it's called cristae. Look at this cristae. This is the inner one. The outer one is the one that in which we store enzymes, proteins, and hydrogen ions. Enzymes, proteins, and hydrogen ion. We store it right here. How about the inner one? The inner one contains matrix. The inner compartment contains matrix, which is semi-fluid. It's not exactly fluid, it's half fluid, semi-fluid, and it contains enzymes. So what do we have in the inner one? What do we have in the outer one? This is something that we need to know, but the most important so far is powerhouse. You make ATP and use oxygen. Here is how it looks like again. This is outer membrane, the orange. The inner membrane is the green. Inside the inner one, inside here is called the matrix, and the matrix contain enzyme. The outer one is the one that also contain enzymes, but contain also protein and hydrogen to make ATP, the outer one. What else do we need to know about the mitochondrion? Important. This is important. It contains its own DNA. It contains its own ribosomes. Very important. But you told us that DNA is in the nucleus and ribosomes on the rough endoplasmic reticulum. Yes, these are additional ones. Additional. So we have other types. And if these types, DNA and ribosomes, we call it mitochondrial DNA and mitochondrial ribosomes. These DNA contain 37 genes, and we inherit it from the mother. They noticed that the DNA can divide independently. Not only when one cell make two cells, so now you have the double of the mitochondria. No, that's not it. The mitochondria itself can double itself within the same cell. So it can divide individually or independently. It's about the size of the bacteria. For those two reasons, there is a theory that's called endosymbiosis, and you guys should know the theory. We talked about this before. Theory. It was chapter 2. So the theory said there's a theory called endosymbiosis. And the endosymbiosis is they said that this mitochondria, it used to be a bacteria, and it got into the cells to give the cells the power to become the powerhouse of the cell. Okay, how did you know that? Like, what's the proof? Because it is the size of the bacteria, and it can divide independently, just like the bacteria. They have their own proof, but this is something that we still need to know. Contain their own DNA, their own ribosomes, come from the mom. Endosymbiosis is, they said, it comes from the mitochondria because it can divide individually, and it is roughly the size of the bacteria.
So it
Go ahead.
was bacteria that altered and turned into the powerhouse?
Yes, this is the theory. Yes, bacteria entered into the cells, crossed the membrane, got into the cells, become modified, and it became the powerhouse of the cell. This is the theory, right?
Does it say what type of bacteria it was that altered? Or
No,
just
no,
in
it
general?
doesn't say. Just one type of bacteria. Okay, so we talked about the mitochondria. What is it? What do you call it? What's the name of or the nickname for it?
Powerhouse.
Powerhouse. Yes, it makes. What does it make? Why did you call it powerhouse? Because it makes
ATP.
ATP. Yes, and it contains its own what?
DNA.
DNA and ribosomes. Yes, and it evolved from ancient
Bacteria.
bacteria. Yes. By the way, if you can answer this, you can answer the questions, because this is very similar to it's not exactly like this. Of course, you put the information that you see here in the form of multiple choice questions, but this is the information that you're going to use. You should be able to answer it for yourself. And also you should be able to read the summary at the end. I don't go through it because it's repeating what we talked about, but the summary and these slides that I just talked about is summarizing what you have to know. And these are the information that will be used in questions for both quiz and exam. Next one is cytoskeleton. Next organelle. What's cyto and what's skeleton? Always do this. Cyto means cell. Skeleton means skeleton. Do you have your own skeleton, like the bone in your body? Isn't that the skeleton? Yes. So why did you call it cytoskeleton? Because it does the same thing. Why do you have a skeleton yourself? Why do you have this bony skeleton inside your body? It is to keep your shape. It's to organize. It's to help you to move. Like I have bone in my upper limb. It's keeping the shape. It's organizing. It's strengthening. Same idea, and that's why they call it cytoskeleton. It's not permanently rigid. It can be rigid, but it can change. We can put it together and break it down assembly and disassembly. It's protein. We make it from protein to give the shape, organization, ability to move, just like your own skeleton. And we have it in three different forms, from big to small: microtubules, intermediate filaments, and microfilaments. These are the three types. Microtubules, this is the largest one. Of course, all of them are small anyway, but relatively, if compared to the other ones. Microtubules, here is the one as you see here, and look at it here, it is maintaining the shape. If you don't have it, the shape of the cells will collapse and it will look different. Like imagine if you don't have the bone inside your body will collapse. Same thing here. Microtubules are the one that's keeping the shape, maintaining the shape inside of the cells. And there is something called centrioles, which is a type of microtubule at the center, and it helps the cells to divide. You don't need to know how, because later we will talk about the cell division. But for now, it's in the center to help the cells to divide. I will tell you how later. Next, which is in the middle, this is an intermediate filaments, looks like this. Look at this picture. It's adding strength. So it's making the cells stronger. It doesn't break down. And it anchors the protein in the cells. So the protein is not like moving around. It's anchored in here. The last one is microfilaments. And the microfilaments also reinforce or anchor, but it play a role, and this is important, in the muscle movements. We have muscles inside the cells, and there are inside our muscle cells. And this is not the time to talk about it, but I'm telling you, when we study the muscles, you will learn that our muscles moves because of these tiny things inside of it. It's called microfilaments. So now we're having a broad idea, and we will move to the details later on. Next, we have also something called cilia and flagella. All the cells have those? No. All the cells have what we already covered so far, starting from the cell membrane to the nucleus, nucleolus, endoplasmic reticulum, rough and smooth, the vesicles, lysosomes, peroxisomes, mitochondria, Golgi apparatus, and all that stuff that we talked about already. Cytoskeleton, yeah, all cells have that. Some cells have flagella, and some cells have cilia. Some cells have both of them, not all cells. These flagella and cilia contain microtubules. What's flagella? Flagella is tail, like this. Like the sperm have flagella. What's a flagella? Translation, tail. And what is the cilia? Look at this cilia, like in the lung, in the airways of the lung, like inside your trachea and bronchi, like inside your respiratory passageway, you have these. What is this? This looks like the brush. You know how the brush looks like finger-like projections. In order to move things around, and we will talk about it more later, but those two contain microtubules to move them. Here is a revision of what we talked about. You should be able to answer it. Cytoskeleton keep the shape of the cells, internal structure to give them the capacity to. So shape is one, keep internal structure is one. The third one is
Movement.
movement. Yes. What are the three types? Microtubules, microfilaments, and intermediate filaments. Some types have some, not all. What are the two that's only in some cells and contain microtubules that we just talked about?
Flagella or cilia.
Yes, flagella and cilia. Very good. And they contain microtubules. Okay, here are other the summary. I'm not going to repeat all of this. It is what we covered from slide 1. These, let me tell you this. After you study the chapter, again, watch the video, read from the slides, and you should be able to answer these. Here's one, and you should know the summary. The summary will give you the vast majority of the questions. Is it repeating what we just talked about? Not exactly repeating, kind of repeating, but summarizing it. So it gives you the basic principles. Here it is. If you go through it, here are the cell theories that we talked about. Not going to repeat, it's exactly the same thing. Here it is, the plasma membrane, the different organelles, but this is giving you the single most important thing about each one of those. If you don't know the summary, you are not going to be able to answer the questions. The summary is the minimum. Here it is.
Just audio.
I'm sorry,
There's no
Yeah,
audio.
same.
I can't hear you.
There's no audio.
Oh,
Yeah, for like the
It's
past
good now.
20 or 30 seconds, it just disappeared.
oh, it looks like there is an issue with the internet. But I don't know where did I stop, but I was saying that you should focus on these, answer it without looking at the next one. If you get a mistake, look at it, do it again. This is one. The summaries. Can you guys hear me now?
Yep.
Okay, so the summaries. I was saying that if you don't know the summaries, you will not be able to answer the questions. So after you're done, you should go through the summary, and it's just going through what we already mentioned, but with a big focus on the main or the most important points. Okay, you should do this and focus on it. You should know every single word here. What should I know about the plasma membrane? The minimum here. Nucleus, smooth ER, rough ER, lysosomes, here it is. Mitochondria, cytoskeleton. So it is giving you what you should focus on. If you're asking me, what exactly should I focus on after I finish studying? It's the summary that you see here. Okay, I'm going to stop and stop